Were there things at the ASH annual meeting that jumped out at you in terms of different potential therapeutic approaches for treating MDS or targeting specific genetic abnormalities? Rami, maybe I'll start with you.
Yeah, I think there are several obviously exciting data. I think maybe I will summarize the APR246, for example. But as you mentioned, I think there are different themes. I think one of them is, targeted therapy. And actually, there is one session that is just dedicated to looking at targeted therapy. So going from a subset of patients that have SF3B1 have good mutation, but like remain transfusion dependent all the time, where luspatercept could be an option, to the other extreme, where patients have P53 mutation, unfortunately have poor outcome, median survival typically 4 to 6 months, poor outcomes with transplant, short duration of response, had been really an unmet need for MDS patients. So several of us had been working with this compound APR246. That's a drug that restores the wild function of P53. It originally was tested actually in ovarian cancer, looking at resensitizing ovarian cancer for cisplatin, because that's part of the mechanism. So we looked at this preclinically, showed some additive effect with azacitidine, did the phase 1 study through the MDS consortium, and at this ASH we'll see 2 phase 2 studies presented, one from our group here and one from our colleagues in France, similar design. Both studies included around 50 patients. Again, those were all patients that had P53 mutation, complex karyotype, and both studies show actually overall responses more than 80%. More than 50% complete responses, and in the range of 18-17% marrow CR plus hematological improvement, and almost half of the patients becoming, NGS negative for the P53 mutation. Again, that's not minimal residual disease, that's clearance of the mutation, because the NGS panel threshold is variable, somewhere 5 to 2%. And then that the median overall survival for those patients at least looks around a year compared to historically of 6 months. Patients that got the treatment, 1/3 of the patients went to transplant and their outcome was better. So it's really very exciting. The phase 3 is ongoing, and hopefully that will confirm those results we are seeing, and it will become available options for patients with really unmet need.
I totally agree. The MDS patients who have a P53 abnormality are just in a horribly tight spot. With the median survival that's terrible. There have been studies that have shown that people with P53 abnormalities seem to actually be really responsive to hypomethylating agents, but not for very long. And that's the dirty little secret about that, right? You can see response rates that may be 60-70% with a hypomethylating agent, but they only last 3 or 4 months. So that may be potentially useful if you're trying to get somebody to a transplant, but not if, for the majority of MDS patients who aren't transplant eligible, for treating them long term. Ellen, are there...
I thought that the CD47 antibody, which is something that you have also been working on at your institution, was also very exciting for the P53 mutated patients. That in the early studies, the patients who were treated up front with this compound, there was a 100% response rate and a very, very low number of patients, but this is exciting initial data, and I think as this goes forward, it will be really interesting to see if this is another tool in our box to be able to treat P53 mutated patients who really had this dismal prognosis with something new. So I was pretty excited about that data. You can probably comment more on that particular trial, Rami, because it was started at your institution, but I think that that's a really interesting drug.
Right. So the anti-CD47 is really a novel way of targeting, macrophages. So it's a checkpoint inhibitor. It targets a receptor or signal that is usually called the "don't eat me" signal, basically evading the immune surveillance from macrophages. It had been tested in patients with MDS as well as AML. Particularly in MDS, in combination with azacitidine, there is really early signal of very promising activity. As you mentioned, originally almost 100% responses, more than 50%, 60% CRs, 1/3 MRD, and we will hear the results of the update at this ASH. So it looks very promising, again, even in patients with P53. So I think that's also being moved forward. The main toxicity with it actually is on-target hemolysis that you see early on. So there is single dose given up early on, because the receptor is on the red blood cells. You see hemolysis, and then the patients will have improvement. So it looks promising as well. And the studies are moving toward phase 3 study with the compound.
I mean, there's also some interesting data in the frontline setting. So, because we always complain about patients who get hypomethylating agents indefinitely, and that oral hypomethylating agents may be the way to go. And so this compound, Aztec 727, we participated in those studies. And what it is a combination of oral decitabine coupled with cytidine deaminase inhibitor, cedazuridine. And so it's a fixed dose, essentially. It's a tablet that somebody can take for 5 days, every 28 days. And the study essentially compared that, actually had 2 different arms, but they were relatively identical, except that with a different sequence of the oral versus IV. And then everybody after cycle 3 received the oral compound. I think that would be really interesting to see if that may take the place of parenteral hypomethylating agents. So that's interesting. And then there's also a combination with pivinitastat and azacitidine in frontline, and there's also in the relapse setting, which will be presented at ASH. So, you know, again, combination studies is very interesting, and we'll see what the future might hold for that.
Yeah. Pivinitastat is interesting. I think I was going to say we'll not leave Mikhail out because he led all the efforts on this. So maybe he will tell us about where we are with the pivinitastat. But like we've had the trials originally, and we saw some promising activity as well.
I'm a big fan of combination studies. The challenge with them is that when you're adding drugs together to an older population, it's hard to keep people on those drugs. And what we saw in the the North American Intergroup study, looking at combinations of azacitidine with lenalidomide or azacitidine and vorinostat, was that docs would lower the doses because they were worried about side effects, or stop one of the drugs, and that would compromise the efficacy. We are seeing promising data with pivinitastat added to azacitidine. It's completed a large international randomized study, also looking at outcomes. So we'll see if those data are positive. What we tried to build into that protocol were stricter guidelines about not lowering doses of the therapy and not stopping therapy prematurely. We really believe that if patients are going to see a response to hypomethylating agents, they need to be on those drugs for at least 6 cycles or 6 months.
There actually was an interesting Chinese abstract that is buried in all the abstracts this year, where they looked at over 1,000 patients, really, at patients who received HMA alone versus HMA combinations in China. And they found that there was a higher rate of complete response in patients who received combinations, but the overall survival was the same in patients who received HMA alone versus HMA in combination. It's hard to know whether those combinations got full dose of those drugs or what happened there, but it's interesting to look at that large population study and see that their overall survival was the same, whether they got the HMA or the combination. So I think there are a lot of challenges when we present combinations to be able to keep those patients on drug at appropriate dosing, and that might account for the fact that the overall survival was the same. But that is an interesting large population study, which a lot of our MDS studies are not at such a large population, looking at the outcomes of combination or a single HMA.
Great point.